Welcome everyone. Today we shall study a new lesson, that is heredity and evolution from class 10. And student, this is part 1 video of this lesson and this topic is from science to class 10. So let's study. Okay, so before starting a lesson, let's revise some information. Okay, so first question is, which component of the cellular nucleus of living organism carries hereditary characters? That means which component of the nucleus contain the hereditary characters. Okay, now hereditary characters means transfer of physical and mental characteristic from their parents to offspring. So so the chromosomes carrying hereditary characters called the genes are located in the nucleus of a living cells. So chromosomes are responsible for transferring the hereditary characters from parents to their offspring or children. And gene is a segment of a chromosome. Okay. So here you all can observe that a cell and the cell contains nucleus, and in the nucleus we have chromosomes, and we all know that 23 chromosomes are there in a cell, and out of these 23 pairs of a chromosome, 22 pairs of a chromosomes are called as autosomes, and one pair of a chromosome is sex chromosome. Okay, so these these chromosomes contain a genetic material, that is DNA. Okay, and DNA, a part of a DNA or segment of a DNA is called as a gene, which is responsible for transferring of hereditary characters from one generation to next generation. Alright, now next question is, what do we call to the process of transfer of physical and mental characters from parents to their progeny? Now progeny means next generation or their offspring. Okay, so the process of transfer of physical and mental characters from parents to their progeny or offspring is called as heredity. Alright, now next question is, which are the components of a DNA? So student, which are they? Okay, now let's understand. The DNA molecule consists of three types of a chemical component. Okay, so the first one is phosphate group, and second is a sugar, or it is a deoxyribose sugar. Okay, or we can say the pentose sugar, that means it contains 5 carbon. Okay, and third component is a 4 nitrogenous bases are there in DNA molecule, and these are guanine, cytosine, thymine and adenines. Alright, so here you all can observe the sugar phosphate backbone, that means phosphate group and sugar group attached to each other, and these are attached to the nitrogenous bases like G, C, A and T. So these are the 4 nitrogenous bases which are there in the DNA molecule. These are these nitrogen bases are attached to each other or join each other by means of a hydrogen bonds in between them. You can observe here that A and T are attached by the double bond, and G and C are joined by the triple bond, and these are nothing but the hydrogen bonds only. Okay, now let's start a lesson. First is, what is heredity? So let's understand. Student, you all know that heredity is the transfer of biological characters from one generation to another generation via genes. So just now we have studied, we have seen in our previous slides that transfer of mental and physical characters from their parents to their next generation or their offspring, it's via genes. Okay, so gene is responsible for transferring of character from one generation to another generation, and this is called as a heredity. And John Gregor Mendel is pioneer of the modern genetics. Now genetics is a study of heredity. So these scientists, that is Gregor John Mendel, is a pioneer of the heredity. Alright, now heredity and hereditary changes. So student, in 1901, the reasons behind the sudden changes were understood due to the mutational theory of Hugo de Vries, and mutational theory means theory of evolution. And meanwhile, in 1902, Walter and Sutton observed the paired chromosome in the cells of a grasshopper. Until then, it was not known to anyone. That means in 1901, first the chromosome are studied in the cells of a grasshopper. and research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes. So in chromosome, we have seen in chromosomes the genetic material, that is DNA, is present, and DNA, a main part or segment of a DNA, that is called as a genes, which are responsible for the hereditary, and through which 1944 trio of scientists. Okay, so the trio of scientists Oswald Avery, Michael McCarthy, and Colin MacLeod proved that except viruses, all living organisms have DNA as genetic material. That means viruses do not have genetic material, DNA as a genetic material, but except these, all living organisms have DNA as their genetic material, which is responsible for the heredity. Okay. Now, in 1961, the French geneticist Francois Jacob and Jacques Monod proposed a model for process of protein synthesis with the help of a DNA in bacterial cell. So, we will study the DNA model of a DNA in our later slide. And it helped to uncover the genetic code hidden in the DNA, and thereby the technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering also. And the science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, and in industrial processes in which microbes are used. So these all are the heredity and hereditary changes. Alright, now main part of our lesson is let's understand the structure of DNA and RNA. Okay, so this is the structure of a DNA in which DNA is a double-stranded, helically coiled double-stranded DNA. Okay, so this is a DNA double helix. That means it is converting like a plates. Okay, so in one strand of DNA is called as a 3' and another strand is 5'. You all can observe in the diagram. Okay, and DNA consists of a sugar phosphate backbone and the 4 types of a nitrogenous bases, that is guanine, adenine, and thymine and cytosines. Okay, and these nitrogen bases are attached held together by means of a hydrogen bonds. So this is a DNA. And RNA, there are 3 types of RNA, that is ribonucleic acid, and DNA is deoxyribonucleic acid. Okay, so the 3 types of RNA is, first is a messenger RNA. So instead of thymine, okay, so in DNA we have thymine, but instead of thymine in RNA it is uracil is there. Okay, and this is a messenger RNA. And structure, you should remember, this is a ribosomal RNA, or we can say rRNA, and this is a transfer RNA, that is a tRNA. And transfer or tRNA is responsible for bringing the amino acid for which is very important part in the synthesis of a protein, complex protein in the cells. Okay, now the process of formation of a complex protein. So in the cells, the protein is synthesis as per the requirement. Okay, and there are various steps are there included in the formation of a complex protein. So there are 3 steps are there. So first is transcription, second is a translation, and third is a translocation. So here you can observe the transcription, translocation, and translation and translocation, just to make the complex protein or formation or synthesis of a complex protein in a cell as per the body requirement of a body. So let's understand one by one this all the 3 steps. Okay. Now with the help of RNA, with the help of RNA, the genes. Now genes is a segment of a DNA. Okay, the genes present in the form of a DNA, that is deoxyribonucleic acid, and participate in the functioning of a cell and thereby control the structure and functioning of the body. And information about the protein synthesis is stored in the DNA. Okay, so the whatever information is required to synthesis or produce or formation of a protein is there in the DNA. Okay, stored in the DNA. and synthesis of appropriate protein as per the requirement is necessary for the body. And these proteins, here you can observe the nucleus and cytoplasm. So in the nucleus we have genetic material, that is DNA. Okay, and synthesis of appropriate protein as per the requirement is necessary for the body. And these proteins, okay, and these proteins are synthesized by DNA through the RNA. Okay, RNA, that is messenger RNA, and this process is called as a central dogma. So what is central dogma? It is the proteins are synthesized by DNA with the help of RNA or messenger RNA. So this is called as a central dogma. Okay, now let's understand the process of first process of transcription. This is our transcription is a process of formation of the messenger RNA from DNA. Okay, so the messenger RNA is produced as per the sequence of a nucleotide on DNA. Okay, so here you can observe inside the nucleus we have the genetic material, that is DNA. Okay, so the messenger RNA is produced as per the sequence of nucleotide on DNA, and for the production of a messenger RNA, the enzymes RNA polymerase, it will first attack the it will first unwind the helical structure of the DNA. Okay, and you can observe here it will unwind the DNA double stranded of the DNA with the help of a RNA polymerase. So RNA polymerase is a enzyme which will attack the any of the segment of the DNA, and only one of the two strand of DNA is used in this process. That means either it will use 5' strand or 3' strand for the production of the messenger RNA. And the sequence of a nucleotide in messenger RNA being produced is always complementary to the DNA strand used for the synthesis. For suppose, that means here in the DNA, if we have the TAC, that means thymine, adenine and guanine sequence. So the sequence for the messenger RNA, it will opposite to the DNA. That means instead of TAC sequence, it will be AUC. Okay, so in DNA, and there is uracil in RNA instead of a thymine in DNA. And this process of RNA synthesis is called as a transcription. That means the process of formation of a messenger RNA with the help of a DNA, it is called as a transcription, and this all process happens in the nucleus of the DNA. So the transcription takes place in the nucleus of the cell. Okay, so the messenger RNA formed in the nucleus comes into the cytoplasm of the cell, and it brings in the coded message from DNA. That means the messenger RNA which is coming out of the nucleus into the cytoplasm, it will bring the coded message from DNA. Now coded message means the three nucleotides together, it will form the one code. Okay, so the message contains a code for amino acid, and the code for each amino acid consists of the 3 nucleotides, and it is called as a triplet codon. So basically the triplet codon will provide the information that which amino acid it required to form a particular protein. And each messenger RNA is made up of thousands of triplet codons, and as per the message, now message means triplet codon. Okay, so as per the message on messenger RNA or mRNA, amino acids are supplied by the transfer RNA. So here this is a tRNA or transfer RNA, it is bringing particular amino acid as per the message, that is as per the triplet codon message are there on the messenger RNA. So as per the triplet codon, the transfer RNA will bring the particular type of a amino acid like lysine and phenyl aniline and methionine. So there are many types, 20 different types, and many more types of amino acids are there which will brought by the transfer RNA to the messenger RNA or mRNA. And for this purpose, the tRNA has anticodon having complementary sequence to the codon on messenger RNA. So here you can observe the messenger RNA and transfer RNA have the complementary nitrogenous base pair. Like on the messenger RNA strand, the codes like AUG, that means adenine, uracil and guanine. So on the transfer RNA or tRNA, the codes will opposite to this. That means it will contain UAC, that means uracil, adenine and cytosine. So this is a complementary nitrogenous base pair. Okay, and this is called as a translation process. Okay, and the amino acid brought in by tRNA are bounded together by the peptide bonds with the help of rRNA or ribosomal RNA. And here you can observe the incoming transfer RNA bound to the amino acid. That means the transfer RNA is bringing particular type of amino acid, that is PHE means phenylalanine. Okay, as for ASP means asparagine, and LYS, that is lysine, trypsin. So these are the different type of amino acid which is brought by the transfer RNA to the messenger RNA. And here you can observe the growing peptide chain. And during this process, the ribosomes keeps on moving from one end of the messenger RNA or mRNA to other end by the distance of one triplet codon, and this is called as a translocation process. So student, these are the different three different steps which are there in the synthesis of a protein, that is transcription, translation and translocation. And such many chains come together to form a complex protein, and these proteins control the various functions in the body of living organism and their appearance too. So these are the three process of formation of a complex protein in our cell. Okay, so student, living organism can produce new individuals like themselves due to the genes only, and some of those genes are transmitted to the next generation without any change. That means some type of a gene, okay, they will transfer as it is from one generation to the another generation without any change. And due to this, some of the characters of parents are transmitted to their offspring, and however sometimes sudden changes occur in those genes. That means sometimes the gene are not passes as it is, their changes will be happen in the sequence of the nucleotide. Okay, and sometimes any nucleotide of the gene changes its position, that causes a minor change which is nothing but the mutation. So here you can observe the two strands, like first is the main or original sequence of the gene, okay, and next is the replacement or mutated part of a gene. So here you can observe the GAT has been broken, or it changes its position, and it instead of GAT, that is guanine, adenine and thymine, it becomes TGC. Okay, so this is a mutation in which the nucleotide nitrogen is or nucleotide change in the nucleotide position takes place. So this is a mutation, and some mutation may be minor but some may be considerable. For example, mutation may cause the genetic disorders like sickle cell anemia. Okay, now sickle cell anemia, so this is you can observe in a small picture, that is normal red blood cell. It is an oval in shape. Okay, so in sickle red blood cell, it is becoming like a sickle shape. Okay, so in normal red blood cell, the flow of blood will be proper and regular, and in sickle cell blood cells, what happened, the flow will be constricted, blood flow will be decreases as than normal. Okay, so this is an everlasting process, and it is one of the theory of Darwin's theory of natural selection. So student, that's it for today's video. I hope you all have understood this topic very well, and next part of this lesson we can understand in our next session. Thank you.